Hello, good morning. How are you? Good evening too. Thank you for listening. This is a medical cannabis show and I respect the law and I hope you do too. The content provided is for educational and scientific purposes, so you can trust what's being said. I fact check the information and the slides that I show are from my university. And those courses are they're written within the last couple of years. So today is an open topic and there's nothing being planned. So waiting for the chat and viewers to put some questions and that will start the stream. If you're rewatching it back, you might want to skip 2, 3, 5, 10 minutes or something and see where there's some slides where the screen's changed, and that's when it will be starting or thereabouts. Yeah, that's about the best I can help you with at the moment because there's no topic. But there's a couple of people here, so g'day to 420 Fred. How you going, mate? Nice to see you, Shipwreck. What up, cheers, he says. So I'll wait till 2 minutes. And then see what I can talk about if there's been nothing that's been suggested. So it's 1 minute 37. Can I talk about a minute 50 now? 2 minutes, I'm going to start talking about something. Come on. Shipwreck and Fred, help me out. What can I talk about? It's 2 minutes. I got to start talking about something or other. My uni started, but I don't want to bore you with my stuff. It's been quite interesting, actually. I've done the course on plant physiology and metabolism previously, but this one was written last year. So it's just good to see the different updates and the different progresses in science that's happened. So it's been pretty good. I should have had 2. If you're watching this back and it goes for 10 minutes, there's been no show. Which is all right. Every now and then it happens. I don't mind at all because it's all about helping the viewers. So if they haven't got any questions or any discussion, that's been good because that means they're happy. So I can be happy too. Oh, hang on. There's a question mark. Shipwreck says, man, it's been so moist here and I've got gifted an outdoor plant. Anything I can do to prevent mold? Oh. Where are you? You're up there. So you're entering summer, autumn. Oh, yeah, so you want to be flipping in about, what's that, 4 weeks? So what I would do, anything I do to prevent mold. Yeah, well, defoliation, get the air flowing through it. The mold spore germinates, the botrytis spore germinates when the humidity around it is above 70 to 75%, so quite high. But if you check the humidity outside just before sunrise, you'll find that most of the time it's quite high. It's around that. And if there's if that spore is not in the wind, like if it's trapped in the middle of the plant, surrounded by heaps of leaves, the humidity might be even higher than 75%, creating its perfect environment for it to germinate. So you've got to reduce that somehow. So, yeah, it should be flipping soon, he says. Okay, so keeping the airflow down and then getting your plant health and high. So if it does encounter a problem, it can activate its immune system and easily disperse it. You can also add some biofungicides. That's really good. And if I was going out into that type of an environment, I'd be putting that in my program. If that's what I was expecting to come out, like in the next month or so, I'd be putting it in my program. So you want the bio-fungicides. So like, I think Bacillus subtilis is even, that's pretty common. Just exactly like that. But you want trichoderma is a really rad one for fungicide. And what's some other really, Pseudomonas fluorescens. That's a bit harder to get. What's easier ones to get that you can, but that's what you want to look at. So the biofungicide, that's what I'd be doing. I got quite a few of the different ones. Metahisium is also pretty rad. Raptor was using that. That's a pesticide, actually. Bacillus, no, Beauveria, Bassiana is really good. Yeah, that's about it. That's all I would suggest. So I'd be going into that environment, keeping all the leaves and everything low so the humidity doesn't get into its germination. And then I would IPM it. It's even said back in the day that milk, believe it or not, you spray milk on it. And that's because milk has a lot of lacto cultures, genus, bacteria in it, and they are heavy spore multiplying. Like they're in competition with everybody else. So they're not really symbiotic. There's, you know, the 6 different types you can get from the microbes. Those lactos are in competition. So you want really symbiotic ones, or ones that don't compete with each other. So they're mutual, co-mutual, those types of things. So by getting the right microbe, you can also counter it if it does fall into those brackets. That's about all I'd go without showing any slides. Do you want me to elaborate more? He goes on to say, Shipwreck says, I'll definitely look into those. I wasn't expecting it, and a buddy gave me a plant that's very close to the size of me, and I'm 7'1". Holy mackerel. He's 6'1". He's not small. I wasn't expecting it, sorry. No, that's all right. Put it into your IPM. What else could I suggest? Do you want me to go to let me know? Do you want me to elaborate more, show some slides, like you can learn a bit about the phylosphere pathogens, the pathogens that are in the leaves above the soil, not the rhizosphere. But yeah, I commonly use Beauveria probably once a week, Metahisium and Trichoderma. I use them at least probably once a week. And if I was going through a period like where there's problems, I'd be using them probably 2 to 3 times a week. Depends on your feed program, because you don't want to overwater. So if it's too wet, you can't give it to it. So yeah, that's what I'd do if I was in that situation. And do prevent mold is the only question so far. Did I cover it? Shipwreck, was that enough? Was that elaborated enough, mate? I could elaborate more if you want me to, but I'm just here to help you guys and girls out. So it's whatever you want. I don't mind at all. But yeah, thinking ahead of your next season and when you're going to be flipping and when your fruit's going to be coming out, like in the middle of that peak time, because that's when everything's congested in the middle. So the bigger the fruiting body, the higher humidity in the center. So if it's only 75% outside in the air, inside that fruit body, it's probably 85 to 90% humidity, which nearly all bacteria germinate at 91%. So it's mega asking for problems. That's why it's hard to grow large fruit. Oh, here we go. Shipwreck says, yes, it was answered. Cool. If you want to go deeper, I'm listening. If someone else asked something, we can flip to that. Yep. I really need to defoliate it a bit. Yeah, especially in veg. Shape it up. Get rid of all the lowers. Get the airflow. Get at least 1 to 2 foot clear off the bottom of the plant, any plant, because you want airflow to go from the top to the bottom, because that's where the transitional period from thermals happen. So if there's if it's hotter at the top, naturally air will flow from the bottom to the top throughout the plant, giving it a gas exchange. So it's good to have those air gaps for the gas exchange as well as your pathogens. Yeah, shape her up nicely. If you want to go deeper, you can. Very true. He says he always defoliates. That's the way. Yes. Well, sometimes you don't have to. Well, defoliation. We can go into I can elaborate a bit on that with the sink and source cells. I haven't really talked about much, but it does get a bit, hey, there's Raptor. Get a Raptor. We were just talking about fungus. You missed out, mate. I can elaborate about the sink and source cells, and then you can learn about why the leaves are green, and why to possibly leave some on. Sounds good. All right, let's go into the sink and source area. Grow plant science, sink and source. Feeding. I'm running low on space, so all the windows aren't coming up, and I can't see what's in the folders. It's kind of in here. There it is, sink and source. I'm sharing, yep. Well, what's this sink and source business? Well, I don't know where this is starting off. The leaf over here, what's this say? It is important to note here that the plants respire all the time, whether it's dark or night. Yes, they do. So once the nutrients come in, they want to store them somewhere. So they'll put them in the sink cells, which is the big massive leaves, and then the source is the new ones. So when the source need different elements to grow, they will pull them from the sink cells. And as long as the nutrients are mobile, there's 3 immobile elements that are calcium, boron, and manganese, and they have to be in the tip. They can't be translocated like all of the other ones, all the other 17 essentials can be. So by taking these off, these are like the solar panel, the storage banks. So just consider that. I know it works really well because there's a lot of growth that can happen, and they do store energy in the roots too as starch, and they can pull on that whenever they want to produce their ATP. Let's just get into this. So that's the general guts of it. So it's the sink and the source, and this will just go into it a bit better. So photosynthesis puts it into, when it creates photosynthesis, transportates via the phloem. It forms carbohydrates out of that and assimilates it into storage. What's this say? Oh, that's just the different elements that are required, the macros and micros, the 3 from the air. Leaves, when immature, are a metabolic sink, while after maturation, they become active photosynthetically and are labelled as source organs. There you go. First strategy, the photosynthesis rate. Carbon translocation. See, I did warn you, it goes into detail. Anabolism and catabolism, meaning metabolism. The sucrose rate. Let's just skip a few through because I can see you going. Please don't go into it this much. Because see, this is how the signal cascade pathway of how it forms. Actually, we're not going to go into that. That's where how it can be stored in the different cells. Triphosphate are transported into the cytosol. That's a bit too in depth. Here we go. This is, I remember, to be a pretty good one. I'm stuck here for a while. So sink, source, and sink. So by removing the big leaves, there's a chance that if the plant isn't in good health, it might not have a few of the nutrients required to complete its growth and development. That's all. So consider that, even though I've had rad success from taking 90% off as well. But this is just how it actually works. Source leaves, they put out messages which transfer all the hormones around. Very good. Seed plates contain. That's getting in a bit of detail. See, I thought this was pretty. It's called callus. Where's the callus? It blocks waterways, and it's a problem. Callose, sorry. It's about the only thing that was really rad. But yeah, see how it's got in here? This is how it moves through the vesicles. Sugars become more concentrated in the sieve elements and companion cells than in the mesophyll cells. Phloem loading can occur by either apoplast or simpler. Oh, gee whiz. That's the routes in which it can travel through the plant. The three ways that liquid can travel throughout a plant: through the apoplast or the symplast, or it can be through translocating through the cells. Pressure flow from the plant. So once the outside leaves respire, there's a pressure created. Therefore, there's a draw upwards, which is here, and then it can get relocated by this pathways that you can see through here. So it gets cycled around in the plant. I'm trying to summarize it, and I hope it's without going into detail. In response to the water potential gradient, water enters the sieve elements and causes the turgor pressure to increase. When the water enters the sieve elements, it swells and the pressure increases. In the entire pathway, where a single membrane enclosed compartment, different pressures at the source and sink cells will rapidly equalize. What's this saying? Symplast. Oh, that's just the different routes. The two, that's showing two routes in which liquid can travel through the plant cells, through the apoplast or through the symplast. So it tells you which way, because that's where the symplast is in here and the apo is outside. And translocating through the cells, like a mixture of the kind of both. I'm trying to get to the interesting bits. Edible sink tissue. Very good. Allocation is key process to sink. Once the transport of nutrients and sugars have been uploaded and enter the sink cells, they can remain as such or can be transformed into various other components. Partitioning determines the patterns of growth. Such growth must be balanced between shoots and roots. And the turgor pressure in the sieve elements could be an important means of communication between source and sink, acting as coordinate rates of loading and unload of the nutrients. What's this one show? The terminal bud contains a shoot apical meristem. Okay, what's the point in showing this? is the sink and source elements. I like that though, because they're all the different individual cells. Pretty cool. It's the pericycle and the quiescent center in there. Anyway, we're not talking about that. Nutrients. Oh, here's a bit of a way how it sort of works. So there's a sources are up here with the big long leaves, and they come in, they feed the sinks when it photosynthesizes, and oxygen comes in through the roots, carbon dioxide goes out, water goes in, minerals, very good. Okay. Translocation of sucrose can be in any direction depending on the source and sink locations and the strengths. The flow of water in plants is almost always from roots to leaves. Isn't it amazing? What is it? 4 millimeters an hour? Hang on. I can't remember it. And it's quite fast. So if you've got a lower humidity here and it's respiring faster, that means it's creating a higher pressure to draw on up from the roots. But if the humidity outside is very high, that means it's not going to respire as much, so it's not going to transport minerals as fast. That's why the VPD comes into it. I don't do that. Okay, that'll do for that one. Too much reading in it. Storage is not necessarily a passive response to excessive photo-assimilate supply. All right, let's get into details. Sink strength equals sink size times sink activity. Isn't that cool? So let's get a bit more in deep into it. Oh, and you can do it by maths if you want. No. Metabolism of the source adjusts to the altered conditions in long-term experiments. That's the through epigenetics. Photosynthetes, starch, sucrose, and hexose. Phytohormone levels in leaves respond to changes in the source and sink ratios. And as you know, phytohormones are like steroids for plants. They're brilliant. Just brilliant. There's no other word for it, because I've been playing around with them for some time. And I'm just amazed at the different results that you can get from it. Like you can, increasing gibberellic acid is similar to CO2. I'm just amazed at the different results that you can get from it. Hang on. Like you can, increasing gibberellic acid is similar. There we go. I'll put it on the other screen just to see if I can see some, oh geez, there's a fair few comments. I'll come back to this. I'll just put it on the other screen. Oh, gee whiz. Hey, it's true, bro. Hey, mate, I haven't seen you in ages. What are we up to? Sounds good. Plants have hormones too. Just talking about that. Yes. I'm getting my microbes to produce hormones for me. How right. Hey, Lene. How you going, Lene? Doing well. I haven't seen Bro in ages. Nice to see you, mate. Yes, it's been too long, mister. Yield source. There you go, yield source. I'll see you, mate. Drop some questions if you've got any. I'm just going on about the sink and source cells, because that's about all we were discussing above. I have some great news to share. Oh, here we go. What is it? Do you want to come on live and tell everyone? G'day, saying g'day. Hello. G'day, GMF. Nice to see you, mate. What is it? Guess what? What's happening? Yield source. I didn't get an email from you. So it must be hot off the press. All right, you tell me before I get back to sharing the sink and source cells, because I don't remember discussing in any of my chats sink and source. So, yeah, oh, righto. He's got some triploid autos. Oh, tetraploid autos now. Seeds are huge. tetraploid. That's pretty sweet. So if you grab the pollen from that, you mix it with any diploid, and you're going to produce triploid seeds, which out of those seeds is the ones that's hard to reproduce. You can reproduce from them, but it's extremely blooming hard. Well, yeah, but that's rad, mate. We have some tetraploid autos. Make some pollen of it. You can still get pollen from your autos. So do that. Yield source, please. That would be very good. Seeds are huge. Yes, that's good. That's the characteristics of the cells, because the tetraploid cells are a lot larger in every way that they grow, because they got to hold more chromosomes in them. So they produce larger everything. Stems, seeds. It's pretty cool. Nice work, mate. It's about time someone blooming fiddle around with that, because that's the future. It's the polyploids, triploids and tetraploids. That's the future. Cheers, cheers. I'll send some photos. Yeah, good. That'd be nice. Tetraploid autos. Yeah, just try and induce them. Put some silver blocking agent onto it in flour to block the ethylene, and then it'll start and produce the male bits. And that's the pollen. And you want those pollen sacs produced, so that'll be invaluable, I suppose. All thanks to me. Okay. Oh, that sounds nice. I'll thank you for that, but not sure how. Anyway, I like to help, so I'll take the praise when it comes. So thank you. So I get back to it. The source and sink cells. There's a lot of people watching. I must be interested in these source and sink cells. He also says the live were shared inspired some breeders. Oh, the live we shared. Oh, that one, rad. Oh, excellent. Cool. We'll have to do an update and see what the latest, see if any one of those breeders might have some questions. That'd be pretty cool. I'll help you out with that. All right. I'll get back to sharing the, yeah, I reckon, yeah, that is pretty good. I reckon it's very, very good. Hello, Matt Holt. If you come in a few times, Matt, I'll and say some stuff that's positive, I'll give you a little wrench thing. Matt says, got a little trouble with purple stems creeping in. This is an interesting topic. Everyone said for years it's been phosphorus, and that's what I believed, because that's what I've heard. I've never done any phosphorus foliar tests to try and eliminate it. But in the latest course, there's an interesting thing to say. It might be nitrogen now. So what I'd do, I just sent this to actually someone recently. Let's see about that. It's a blimmin', okay, it's just a lecture. It's not the actual, like a snippet of when it actually happened. I wonder if I could even play it. But it's fascinating. So make up a 100 ppm mix of ammonium or nitrate in foliar and give it to it and see if that works. That's what I suggest. If you do really want to learn about it, Matt, I'll share screen then. This is the lecture. So it's the role of nitrogen in plants, and it comes up in that. So I suppose I could share this. It's the private lecture. It's only written last year too, so it's all up-to-date information. This is in the new course that I'm doing. How can I get this? Go to my playlists. I'll make the playlists on this course available, and you'll see in here the answer that I'm talking about. And it's, yeah, it's from the Aligarh University in India, from the Department of Botany. So it's written recent. So that's why I say to that one, I'll stop sharing now, which is pretty blooming interesting. So yeah, give that a go and get back to us, Matt. You lurk, no worries, mate. That's cool. Interesting live tonight. I'm listening. Oh, okay. No, it's only interesting if people can give me some questions, and then I can elaborate on that, because otherwise I'm thinking I'm speaking to me and it's not as interesting for me, so I can't get into it. Where if there's a question, I can really hunt it down and elaborate around that question, and then the person who asked the question can make up the answer for themselves type thing, because they'll have all the information. But yeah, thanks for that. Nice words, mate. Nice. Cheers. Okay, so that's, any other questions? Come on, pump them out for me. So I've got to put that one, I'll unlock that playlist, and let me know, Matt, if for some reason I forget, please. Yeah, because it does happen. We've got a lot going on at the moment. Back to the phloem, sink and source cells. Yeah, all right. I've got the tablet open so I can see some questions that come through. Here's your phytohormones. You're talking about, Raptor? Phytohormone levels in leaves respond to changes in the sink and source ratio. So once you go and cut down, do a massive defoliation, the ratio is going to change with your phytohormone amount in the plant. And how does that change? Well, the gibberellic acids did not stimulate leaf photosynthesis or alter photostimulate partitioning, or appearing instead to upregulate phloem loading. So it affects gibberellic acid. So that's why it doesn't stretch after you do a defoliation. And alterations in sink demand and in the sink limited system elicit immediate effects of the photostimulate export. Okay, good. Source activity, here we go. Photosynthesis, nutrient remobilization, export, germination. So what's this really saying? Sink activity is low with sugars and can be translocated high. Right. Source activity is low. That's the opposite. Source and sink of the sugars. Okay. That's just showing that. Vascular tissues. All right. Do you want me to, what do we do? Sink and source. Well, it's talking about the phloem and the and the xylem. The xylem's got two. Don't want to really go into that much detail. The tracheids and the vessels, and then the phloem has got all these little other bits. It's pretty rad. Being a dicot, if you cut the cross-sectional area of the stem, this is what you'll generally see. Little holes and stuff in it like this. That's a blown-up version of it. Basic plant, that's where it would have been good to start. Plants have 3 vegetative organs: roots, stems, and leaves. Signal transduction pathway of what? Light. Oh, that's working. Photosynthesis. The different fibers that are in the walls of the plants. It's pretty cool, isn't it? And you can adjust them differently. You can like faster growth will stretch them more and make it more hollow. Slower growth will fill it in more with wood. So you might get to see the different styles of stem. That's the smallest motor in the world for the No. It's producing ATP, phosphorylation, photophosphorylation. Isn't that right? There's the rotor and the stator, and it actually spins and generates energy at the top. Going a bit off track here. Transcript. Oh no, what is that? Oh, that's it. Okay. What's in there's some cool things in this week's slides. I'm just going to pull up that because I remember some cool things for not this week, this latest course. Study. What's the name of that course in the plant? Yeah, it was about water retention, mineral nutrition, and classification. So the water retention does kind of... Oh, there's a question mark. GMF says, I am dog sitting a small pack, and it kind of blows my mind that these puppies were all modern people, the equivalent of hype crossed to hype polyhybrids. Are modern people the equivalent of hype crossed to hype polyhybrids? Modern people are a weird species. A lot of inbreeding happens, and there's a lot of spacism. I prefer, actually, I'm not going to go on, elaborate about that. I prefer, GMF goes on to say, for modern plants, I am thinking it is about the same, and people are smarter, bigger, and stronger than ever before. People actually, I think their intelligence has gone down since the internet's come in because they didn't have to retain as much. Something along those lines I remember reading. But society's just weird, isn't it, mate? Yeah. You can relate plants to humans because we have got the same type of cells, the eukaryotic cells. And we do go similar, like we activate our immune system for problems. We've got a metabolism. It is pretty similar. We use nutrients in different ways to get our energy. Oh, that's right. I was getting to this one because I thought there was some interesting things that related. How do plants absorb water? Oh, no. I found this interesting, actually, because the solute having a solution going into a plant. I found this interesting, actually. What happened here? Because the solute having a solution. What happened then? It just started playing for some reason. Oh, it's a weird computer. I didn't know where old Flamin' was. Having a solution, like if your ppm is got a reading on it, that means it's not pure water. And a plant having solution in it will push the solution through the phloem and the xylem actually slower. So if you've got a really hot day outside, for instance, and you want your plant's respiring and wilting lots, you want it to retain the moisture. So by giving it a solution with a higher ppm, it will not get it through the plant as fast as it could. That was an interesting point that I learnt from this lecture. What happens once water enters the plant? It goes through those three ways. Remember I was talking briefly before about it? There's the third one, transmembrane. So it goes through the symplast, or through the apoplast, or through the weird-looking transmembrane way. The barrier, this gets way too technical. Translocations beyond the epidermis. Yes, 98% of the plant gets its water through its roots. 98. So 2% can come through the dermal cells. The dermal tissue. So in other words, foliar spray works at 2%. This is pretty interesting about how the water and root pressure and water can actually get the water. How does it get it right up to the top of these sequoias? These 100-meter, 300-foot things. So yeah, I think I've touched on that, cohesion and adhesion, cohesion tension theory. Cohesion and adhesion. It's a different theory again, because water molecules, you know, they stick to each other, so they want to keep joining to each other, so they cohesion and they adhere to each other, sort of doing a pulling thing. And once the pulling thing stops, it gets air bubbles, and that's called cavitation. And the air bubble is an embolism in it, and that's where you get problems. Like when people clone, they say, don't get the air bubbles in it. Well, it's got to be kind of photosynthetically, cavitation. There it is. So that's basically how that happens. The gas is trapped with the air bubbles inside of it. Not good. Some rad things. How it respires. What was that interesting point? Through the xylem at the rate of 4 millimeters per second during fast evaporation. So isn't that a spin out? And that's from the Plant Physiology 6th edition book. 4 millimeters per second. I find it a little bit hard to believe, but in other words, it can evaporate extremely fast in perfect conditions. With low solution in the water, it can just be pumping it out. Different types of transpiration. Yeah, there's 3 exits. Through the lenticels, which are mainly in your birch trees. You see those little slits on the side, that's the lenticels. They're like a stoma on the, which is multiple stomata, is called stomata. It's like another breathing point. And a hydathode are at the ends of the leaves, where you'll see droplets occur, which there was right ages ago. There, right on the leaf margins, that's where the hydathodes are. It's a bit about water today. Any other questions? Oh, there is a little bit. I think so hot on it. Pugweed, I think so hot too. Good info here, Aussie. Oh, thanks, Shipwreck. Shipwreck says, I have a few plants push out their sugars over the years. Oh, yeah. If you taste that, you can get a little, just a mild, the phloem sap, its constituents. I saw that not long ago. Gee, it's hard not having these pictures up. That's a shame. I was just going to read to you what's in the phloem sap. Oh, not going to. Factors affecting the rate of transpiration. So if you want it to transpire slower, how can you slow it down? Or if you want to speed up, how do you speed it up? Well, temperature. It goes faster with higher temperatures. Wind. It increases with wind as well. What's this say? Stops at high wind velocities and increases during low. It's because it stops, it will block off the stomata, they'll close. Oh, here it goes. Stomata close at high wind velocities. So no transpiration occurs. And light is another way to affect it. Increases the bright light. The stomata open wider and allow gas exchange for photosynthesis, therefore allowing more water to evaporate. and the humidity, a decrease in the rate of transpiration. So leaf is already surrounded by moist air. So diffusion of water out of the leaf slows down with higher humidity. So there's some ways you can manipulate the plant, for instance. It's really good maybe if you want to, at the end of the growth cycle, you might want to flush. So you might want to get rid of excess and make it transpire quicker. So if you want to slow it down, plants employ various strategies and adaptations to reduce the loss of water, like sunken stomata. It'll be just, but if you put it under a microscope, you'll see it won't be on the outside. It'll just be a little bit recessed inside a little bit. And they have a thicker waxy cuticle. So on the outside of the leaf, there'll be on the dermal cells there, it'll be like it'll look real waxy and shiny. And that's because it's built up a thicker waxy cuticle to protect it from evaporation. And the third one is the presence of stomata on the lower of the epidermis. So instead of on the lower leaves, they go on the lower epidermal cells. Anti-transparence. So there are chemicals which are used to reduce loss of water from plants through transpiration. And there are basically 4 types of anti-transparence. Stomatal closing ones, film forming, reflectants, and growth retardants. Stomata, the green gatekeepers of gas exchange. Actually, this is a better one because that's a monocot. See, it's corn. It's a monocot, so it looks a bit different. So this is a dicot. What causes opening and closing of the stomata? How do they open and close? Well, there's a few different ways, but this is just to talk about the potassium and chloride coming into it and increasing, and it makes it go straight and closes it out. Sorry, when it's the opposite. See, over here it says closure. When it leaves, it straightens it out and closes it off. Because when it opens it up, it fills up and it forms this, it goes rounder and rounder. Stomatal opening and closure. Also high CO2s, quite a few things. Actually, I think it comes up to that and tells you about that. Factors affecting stomatal movement. Heaps of things. Light, temperature, humidity, atmospheric humidity, CO2, oxygen even, water availability, pH, growth hormones, and mineral status all affect the stomatal movement. But what happens? The temperature above 35 C induces stomatal closure. That's why it's important to have canopy temperature as well as air temperature. Closure, while high humidity promotes the stomata opening. And space caused stomatal opening during the night. What's this one? Cytokinins are well established to stimulate stomatal opening. Oh, wow. Forgot about that. Induces stomatal closure. So abscisic acid is another plant hormone that does it. The closer to close the stomata, helping to reduce water loss. That's why it will close it. Okay. Mineral nutrition. This goes into something different. Good. That's it. It says water. A brief introduction on water. So I can really, I got stacks on water. But I want to talk about something about water. Got 10 minutes. I could elaborate a bit more on water. What do you reckon? You love sequoias. Yeah, they're humongous. All right, let's see what's in my water section. Water. Aquaporin, root pressure and guttation. Water relations in plants. We're sort of getting a bit down this route. Let's see what's going on here. Fair income. This is where it shows the phloem sap. These are the different theories that we're going through a plant. Okay. And so you can test it. It's pretty rad. Put a pressure gauge up to the stem and you can test the pressure increase. See how much it moves. It's pretty rad. I like these experiments because you can do them yourselves and you can see, get the answers here. Like that one. A glass tube over the stem with a rubber a bit around the membrane, making it airtight. And then you can see from the root pressure to prove root pressure theory. So root pressure is the transverse osmotic pressure within the cells of a root system that causes sap to rise through the plant stem to the leaves. Don't want to go into detail. Of course, root pressure occurs in the xylem of some vascular plants when the soil moisture level is high either at night or when transpiration is low during the day. That's why it's good to give your soil moisture just when the sun comes on, sun comes out. Cause of root pressure, no, I'm not going into that detail. Significance. Root pressure can transport water and dissolved mineral nutrients from roots through the xylem to the tops of relatively short plants when transpiration is low to zero. In modern concept, no. Use of root pressure cause of guttation. Sort of said that before about the leaf margins. Oh, here we go. It's the exudates, not the phloem sap. Actually, I think I've got the phloem sap too. Anyway, this is the exudates. So guttation fluid may contain a variety of organic and inorganic compounds. So at the edge of your leaves, you'll see the little droplets of water when the humidity is too high and it can't respire properly. So in other words, that water can't evaporate. It just sits there and builds up on the edge of the leaf margins. And inside that solution, it's made up of minerals such as nitrates and salts of SP, magnesium, manganese, sodium, aluminium, fluorine, hexoses, and even enzymes like catalase, amylase, and peroxidase. On drying, a white crust remains on the edge of the leaf margins. You can see it. Depends what type of chemicals are in there. If it was high in sodium, for instance, it might leave your white crust on the edge. And yeah, that'll do. But that was pretty good just to get the answer out finally. Yes, your hydrophones, very good. Oh, there you go. The guttation. A good way to see if there's enough root pressure when you've got a cloning box is that you'll see water droplets on the outside. That means that there is sufficient roots and the plant should be removed. There's an example of guttation. Not looking too healthy, is it? Anyway, look, there's even a bit of fungus there. Maybe this was an experiment, but still, that's guttation. That's all right. That's just the one leaf. So that might have had some droplets on it. So that should have been cut off. Yes, because see, notice how there's not guttation on this leaf because it's not healthy and there's been some dead tissue. So there's no action going on in that tissue, and the cells that are beyond that, it can't transpire or move through the vesicles. So it's just there's no action. So that's why at the end of the leaf margin on here, you don't see any. And on the healthier ones, you'll see them. They're pretty big. So the humidity is too high, or the root pressure is too high. So yeah, the humidity is too high, you can tell that. So it needs to respire. And that I like that because it's just out the cut petioles, just where the stems, leaf stems were, and it's the root pressure is just causing it to sap out through there. It's a bit healthier. Still got, looks like it may be a nitrogen, start of a nitrogen deficiency. There's a good one. Oh, isn't that beautiful? It's got good root pressure, isn't it? Is that all? Root pressure and guttation. Water relation in plants. Oh, Genetic Memory Farms has got a question I see. Oh, okay. He's got the first one to shipwreck. We can't really say of these people are tall and whatever because we have significantly scrambled the pieces geographically population of people and less homogeneous people like them to be freaky. Same with plants. He goes on to say, I guess I am talking about landrace populations with modern diasporas. Yep. Weirdness. And then introduce all of the different microbes. So remember, if you go to the landrace folk that's in Africa, they've got the landrace generations that still live in the land. They have to hunt every day, and they did a fecal transplant, and they found out that their microbes in their guts were phenomenally rad, and they could never get cancers, all that sort of stuff, because they've got the right microbiome built into their system from their practices that they do each day, keeping healthy, running around, getting their food off the land. And us, the modern human, is not as good. That's why it's all cancers. But anyway, that's a different bloody topic. Today's the water topic. Poor humans. What else can I show for water? We've got another 4 minutes. Put your questions in now if you want, because oh. Oh. I'm not sure I am sharing. Water relation in plants. This might go a bit too much into detail. For 4 minutes I wanted to cover. Actually, that's a good idea. I'll stop this one. And go into this one. Water stress. Let's start at the back. Well-watered, water stress. What's the difference? Well, you can see the stomata is closed on water stressed. Seed size is heaps lower in the yield. Photosynthesis is way lower with chlorophyll. The stem, the roots is way longer because it's tried to find a source of water. So instead of putting its growth and development up in the phytosphere up here, it's put it down below. So it's massive, massive importance for water. And it's really hard to not overwater or underwater. So by knowing your permanent wilting point is so beneficial. Just when it starts to wilt, that's the maximum it can handle, and it's starting to drop its water in its cells. That's why it goes limp. So then you've learned, all right, that's the actual maximum I can ever push it to, because beyond that, it starts to do bad damage. It starts to produce abscisic acid, and it starts to induce not the growth and development. It goes into the stress phase, like on this side. So, yeah, that'll do. Next, basic functions of water. No, irrigation use efficiency is so important. And now they've got a thing called nanobubbles, where the effects of nanobubbles of improved irrigation efficiency. Biofilm clogging reduction, reduce water tension, improve soil fluctuation. Fluctuations more efficient water and nutrient uptake. Results of scientific trial. So yeah, that's something that's come out of recent. Nanobubbles. Free water content test. What's this? Water moisture oven test. Oh, okay, that's to test your moisture content. Very good. If you're interested in testing moisture content, that's how to do it. Hey, I love that photo. Isn't that rad? Apoplast. There are the 3 ways it can move the water through the plant. Very good. Didn't see that callus. Transpiration. Oh. Optimum soil temperature for maximum rate of water absorption is 20 C to 30 C, or 65 F to 80 F. Soil aeration. Water absorption is done more efficiently in well-aerated soil. If the soil water increases much beyond field capacity, as happens during flooding, air pores are clogged, and remember the plant is 10,000 times slower to get oxygen out of water than it is out of the air. And also water absorption stops. That'll do. Without going into much more detail, factors affecting the rate of water absorption. I'm going backwards, so it's not getting to a point. Well, 95% of the water absorbed by root system is operated by passive forces, like the DPD gradient created by the transpiration pull. Sort of covered that as well. What's this? Roots take up more water. Water is pulled up through the xylem. Water is replaced from inner cells, and then water leaves through stoma and evaporates. Perfect example of absorption. Pathways. Okay, we've been through that multiple times. Soil water. Soil water is 5 different points it says here. Gravitational water, capillary water, hydroscopic water, runaway water, and chemically combined water. And that's all very relevant if you're feeding your soil, because they're all different processes that happen that you want to know about to maintain equilibrium. Where's the big water thing? Oh, this is a good one. So this is 10 meters or 30 feet vertical distance. The water translocates into 0.1 megapascal change in water potential. So that's just saying the water gets up there with the change of water pressure, water potential. What the big square is. There's an example of a turgid cell with a good amount of water in it, maybe a bit too much, but this is flaccid, and see why water loss by exosmosis, vacuoles shrink and cell loses its shape, leading to the loss of turgor pressure and wilting of the plant. So it's not healthy, and that's where it's stressing. You don't want to stress it out. Oh, yeah. Same as plants. Hang on. Raptor says, I've noticed guttation after a defoliation of my plants. Oh, really? I've seen it in person on my strawberry leaf too. So you've noticed a guttation. Wow, that's interesting. So that must mean that you had too much water in your substrate. So you could, next time, you could make sure you do defoliation when there's not as much water in the substrate, possibly. Actually, but, or have a lower humidity in the air, which would be better, because you want the plant to be doing its transpiration. You don't want that process to really slow, I suppose. Or you could in different circumstances, like if there's disease or if there's a problem and you don't want it to plant, because if you get bugs in the room or in your plant area, there's phloem suckers. They get into the sap, so they'll get in, and that will move the fluids around your plant faster, allowing the disease or pathogen to go to different parts of the plant. So you might want to slow it down. So it's a way when you could slow it down. Shibrik says, Granny growing her, oh yeah. Water, righto, let's get into the maths bit. Without getting water potential as reference state. Righto, very good. I think I've touched on it all. Put stuff in the chat, please, because it might finish soon. 6 oxygen is Since oxygen is much more electronegative, its atoms tend to draw the electrons away from the hydrogen atoms. Okay. Oh, well, we're not going into that much detail. Imbibing. Under water stress, plants produce smaller pubescent leaves to minimize water loss. Ah, that's something you can notice. Underwater stress. Oh, wow. So if your plants aren't as big. What's trichophily? Wow, the presence of trichomes or hairs on the leaves or stems. Trichophily. Leaves rolling, shown by grass leaves during water stress to minimize transpiration. Yep. So, you know, it's called like a canoeing effect even. There's your thick waxy cuticle, which you don't want really, because it's going to minimize the amount of moisture that can be absorbed and released through the epidermal cells here. Yeah, that's what it looks like. You've probably seen them on like fruit trees. There's the hypodermis, sunken stomata. So see how it's outside. It should be out here, waxy cuticle, the epidermis near on those outside, and it's right back inside here. So it should be up here and sunken back in. It's one of those examples before it was saying signs of H2O stress in leaves. Read those out before. Thick epidermis and hypodermis, bulliform cells. Drought escapes is the ability of plants to complete the life cycle before severe stress sets in. Showing that. Is it an example of water stress? Let's see, little miniature leaves. Not very good. Mild drought stress leads to tenfold increase in the endogenous abscisic acid concentration, both in the case of higher and lower plants. So on all plants, a mild stress leads tenfold increase in the bad abscisic acid, which tells all of the plant cycles, it's one of the plant hormones, and it tells all of its cycles to stop growing, stop developing. Gee, what's going on? Quick. Drought stress causes signal perception followed by signal transduction that results in gene expression. Okay. Water scarcity is one of the major limitations for plant productivity and elicits a range of responses at more different stages. Okay, that'll do. For water at 25%, okay, the heat of vaporization is 44 kilojoules of energy. Right, that's it. I think I'll be boring you guys and girls. I've been for an hour. 10. Very cool. Yeah. Well, put your stuff in the chat. That's it, I reckon. I appreciate everybody for listening. It was really good this week. So what can water? It can be retained to phloem and sink and source cells, importance of water. What else was there? Plant reactions to water through the stomata example, as it closes and opens and things like that through different light and different. So I reckon that can be today's topic. Brad, good stuff. What water? It's got what plants crave. Yeah, water, it's actually 70% of the plants, pretty similar to humans. The amount of water that it retains in the body, retains in its cells. Good stuff. Yes, I think so too. If anybody wants to learn more about the water and why leaves are important, yeah, I think it's pretty good. Yep. Thanks for another great show. You're welcome. Oh, here we go. Question. Lene says, I'm off to catch the sunset while I can. Oh, happy sunsetting. I hope there's a good, nice, some clouds in the sunset so you can get some color action. Generic Memory Farm says, question. I'm finishing a cherry Malawi and it is a sativa that can be so much. So I think that senescence is different. What are your experiences? should let them dry out at to harvest. Citrus sativa, so much so I think that senescence is different. Yeah, it lasts longer. Depends on what is it from? Malawi. If it's more equatorial, it'll have a lot different traits. It'll have the traits allowing to the equator, where it might go for a lot longer, and therefore the senescence would go for longer. So if the normal flowering of indica, for instance, might be 60 days, sativa might be 90. So you might get senescence for an extended period. So yeah, I think it would be a lot different, mate. My experiences haven't got much in that area. Don't play around with those ones. Should let them dry out to harvest. I think that's, should the plant dry out upon harvest? Yes, always. Oh, actually, well, not always, because you can just, it just means it's just going to dry a bit, it's going to retain a lot more water, so your drying mightn't be as fast as it normally is. So just have to take that into consideration if it's not dry upon harvest. I hope that helped. Let me know if it didn't help, mate, in the questions. Raptor says, enjoy the Perseid meteor shower, everyone. Oh, really? We've got a meteor shower tonight. I'm glad you said that. Thank you. I'll be looking out the window, Raptor. Everybody else too, check out the window tonight. Oh, yep, sounds good. Hope everyone has a good weekend. Says Lene. GMF says, none of the hairs turned orange. Super thin leaves. Big bushy. Oh. Well, that means it's still going. Let it go, mate. Let it go. It's still, you might have some genetic traits that extend it. You could have some blocking of the different hormones that come out. You could have an increase of cytokines that extend it as well. So there could be a few gene reactions that are happening there to get that result. But as for what I understand, it's got a lot longer to go. You might have a 120-dayer, which you'll be wanting to cross with ELF dominant gene to reduce that. Raptor says it's Sunday, but Sunday is the meteor shower. Get ready, everybody. Get your helmets ready. Raptor said, no, he didn't say helmets. He just, I said helmets. Shipwreck says, sounds like a, yeah, it's those hazes. Whoops. So not good for reading. Yes, write it out. I've, as a general rule of thumb, where is it? The plant maturity is when the anthers or those pistils turn amber or they start and go red. If they're still white, they're still in their growth period and they're not finishing up. So I've had a couple of things like that in the past and I haven't been too impressed with it. And it's just finish. Actually, I've got a video on that, on how to extend, where I took a normal 60-day to 100 or 110 day, and it was just still just going and going. The fruit was the size of the elbows. It was yeah, it's just, the bigger it goes, the more problems you're going to have too, and all sorts. It's another sort of story. That's why it's good just sticking to the 3-4 finger wides, I reckon, instead of the arm wides. In my opinion, that is. Yeah, write it out. Yeah, mate, write it out, and even grab some pollen and try and whack it on one of your not autos, but you don't have any ELF, early flowering gene. That's the name of that dominant gene. Sorry about that. But another comment. Yes, you'd love that one. GMF, it is already 11 on, 7 off, 11 on. Huh. That's a pretty cool. Ask me about that one next week, GMF, about the different hours for the outcomes that you can get. I was very surprised in 10 hours on, 14 off, how well that did with no CO2. It's a bit of a trick for manipulating tightness, and it can be done outdoors too. It's pretty cool. Where I always thought, no, you got to go nearly the opposite, but yeah, it's pretty rad. There's a good paper written on that. Raptor says, if you can reduce the light intensity and temperatures, that can help with density and senescence. Oh, okay. Raptor must have done it. So, well, if you want to increase senescence, add ethylene. That's promotes senescence, leaf senescence. It ripens fruit faster. If you haven't got ethylene, like bananas, or you only need like a big bunch of bananas too. You can use kerosene, because the kerosene heater, the molecule is very similar to ethylene, and are finding the back of the early 1900s, the street lights are finding why with certain plants, with certain trees, senescing quicker, and that was the reason why. Yeah, a good little comment, Raptor. Just to add in, if you want to finish early. Yes. How else can you finish early? Well, by reducing the nutrients. If you sort of starve it to death. Oh, look at these. Where did all these comments come from? 11:13 might be normal. Okay. This is the first run. Oh, yeah. Yes, indeed. Oh, that meteor showers in the northern hemisphere. Oh, so you southern hemisphere folk. Dun dun. Most of the people here listen to shows in the northern hemisphere, though. Say 90% virtually. And Renee says, awesome. Look forward to hearing it. More like look forward to, yeah. And I got to put that playlist. All right. That's about it. AussyCC. Oh, yeah. Wash and hang a few apples. Great idea. Yep. And even cut the apples, because the cut fruit releases more senescence, more ethylene, sorry. So if you go around and put like little slices, say if you have an apple in your hand, you might want to put, say, 6 or 8 slices from top to bottom that go in 1/8 of an inch, and that will increase its ethylene release heaps fast. Or even, I suppose, depends how long you want the ethylene to be around for. If you want it to be around for a week to 10 days, you might want to only cut 1 or 2 sides, because it'll brown too fast. You know, vinegar stops browning. You know that, though. But if you want to do it faster, if you want to sort of finish within a few days, like 3 or 4, you might want to cut it up and then release it, and you'll start to see a really fast senescence starting to happen. If you've got the right amount of ethylene, I should add, because it's a ratio thing. You can't put 1 apple in to a large area and expect action. You know that. Just wanted to clarify for those who were going to do it, though. And bananas. And what's the third fruit that, well, as it's aging, it releases ethylene. It's volatilization. If you look in that, pears, I think it's the third one. There's 3 that are really, really rad. But I use apples. I've always got a few apples. I just permanently just leave them in the fridge. Although ethylene helps with seed germination. it's another reason to keep apples around. Shipwreck says, I definitely want to hear about that cherry Malawi. Geez, you're getting everyone excited, GMF. Seems like you're going to have to make some seeds, Nelly. Righto. Thanks, everybody, for rocking up today. I'll leave it 30 seconds after again just to put in any comments. But I'll see you next week. Bring your questions. Thanks for the excellent show today. I really enjoyed it. Really enjoyed everybody's company too. It was great. Great fun. Good crew. Always got bananas. Should I try it on my 6.5 week? Oh, well, it does work. Raptor, it dead set works. So if you want to finish a bit early, remember the bulking stage happens at the later period because it's going through a sort of a 40-day flowering, and if you cut it a bit early, it misses out on that last bulking phase. So it's up to you, mate. It's up to you. Now that you've got all the information, you'll know what's going to happen if you do it and stuff, and if you don't want to do it and blah, blah, blah. Maybe you might want to do it if you run a bit low on your meds and you want some, but yeah. Good stuff, good comment. Good stuff, says Shipwreck. Thanks, mate. Hope everyone has a good weekend. Good on you, Shipwreck. Yes. Happy breeding, happy growing, and good health to you all. I'll see you all next week, same time, same place, and bye-bye for now. Only have 6 seeds. Ha ha ha, very good. Holy mackerel. That's all right. All right, bye-bye.